So good morning. Thanks for having me. So again, my name is Matt Kramer. I'm the National Sales Manager for Kaijin's applied testing business. So what that is at Kaijin is I have responsibility for our North America business for veterinary diagnostics, forensics, biosecurity, and anything you want to put into the loop of biosecurity. So I'm going, for instance, right after this to CDC to talk H1N1 with the CDC. So I do quite a few different things. I joined Kaijin in 2003 as an instrument specialist. So I would say ultimately that's my background is automation. From there, I joined the applied testing team when it started here at Kaijin in 2005. And from there, I've come to run the business for North America. I am a chemist by background, so I do know a little bit about DNA, but no, just kidding. I've certainly coursework allowed for specializing in both bio and chemist, but I am a chemist by background. So sometimes it's a different way of thinking than biologist, and so sometimes it can all work out well for people. So. At any point, I guess we'll probably take questions at the end, probably work out best for everybody. And, but please do, if there's something, particularly up on the slides, please don't hesitate to ask me a question. So with that, I'll start the presentation. So again, the purpose for today is to really introduce you to the idea of post-PCR cleanup as a forensic technique. What I want to make sure I do is to not get into too much detail, because Rob will really be going into that throughout the presentations as well. So I'll try not to get into too much detail, but to really to describe more around the concept, what we tried to achieve with post-PCR cleanup. So, so why? And one of the main objectives here that we'll try to answer here is why use this technique. The second will be to the whole just idea now that we have around increased sample throughput. So we have more and more touch samples, more and more specialized samples. The expectations are increasing on the DNA forensic laboratories. How do we handle bone? How do we handle a small piece of a hair shaft and get good results from it? Taking those specialized sample types and making them routine is what we really want to achieve. And so that really affects the way also that we need to think about throughput and triaging samples, and we'll talk a little bit about that. And then most of all, and really where Kaijin really, I guess, relies upon is our customers. And so we'll be going over some customer data today, a couple different studies, and really what we do, Kaijin is a extraction and assay company. We obviously, post-PCR cleanup is nothing new to Kaijin. We've been doing it basically the whole human genome, I would say, was provided to us through post-PCR cleanup. So if you go back and look at all the write-ups, I think pretty much every customer was using QIAQuick or some sort of post-PCR cleanup when providing the whole genome. So it's certainly been a application that's been around for a while. So again, one of the things and why do we want to do this again is really the whole idea of this increasing samples. The labs, again, you're being asked to do more and more samples and more difficult samples. And so this you'll see is a tool that we can help do that and manage that throughput. Again, a lot of laboratories, we also have to talk about and think about how to triage samples. So do we have a specialized way that you want to work possible LCN, low template type samples versus high template samples? And so that is one of the things that we do have to think about within the laboratory, is not only just learn about it, figure out, Rob's going to talk about how to implement it and how to work with it in the laboratory, but then how do we deal with it in the laboratory? And we need to think about that process as well when you go back to your laboratories. Again, in terms of ways and applications, a lot of people think about MinElute as only a low copy number type, technology, or its application of. But I would say that there's been applications where there's just, for some reason, the amp was dirty amp. There's just something in it, some high salts are left over. Also, we also see that a lot within, say, traditional, so Chelex, phenylchloroform type extractions. where the application of MinElute can be done, let alone if it's LCN, low template amount. You know, we really need, there's other ways and other reasons to use MinElute, as you'll see within some of the data that we've seen as well. And then again, you're being asked to do more and more specialized sample types. We've also seen application where MinElute is being used for degraded DNA samples. So instead of utilizing and going through the processes of using new technologies like, say, MiniFiler and some of the other new Mini-STR technologies, you may be able to implement and to utilize MinElute as an alternative to doing a secondary prep instead of doing Identifiler and MiniFiler. It may just be a way to implement that. Again, there's various techniques to handle these specialized DNA applications, as you know. So again, low copy number, low template PCR. Again, we'll talk about in some of the applications in the studies here, the benefits and the costs of doing some of those, that LCN type work. Also, again, alternatives, MiniFiler for degraded DNA. Whole genome amplification, we're not going to talk too much about it today, but again, that would be a different concept for enrichment of DNA. So we would enrich the entire genome and then do a PCR amplification for our specific primers. So there's different alternatives that we can look at. Again, post-amp cleanup, and then also other alternatives would certainly be mitochondrial and then the YSTRs. One of the things that we do see is really the application. If you look at the Mann's Press, her secondary paper on the postcoital research that she did, they actually coupled those two, so doing post-PCR cleanup and the YSTRs. So again, a lot of these technologies and applications can really complement each other. Throughout some of the data, and really a lot of our customers who have validated MinElute already, one of the things that they, as they went through the validations, were really trying to keep an eye on was allele drop-in and drop-out, increased stutter, the heterozygous peak imbalance. So those are the various issues that we're trying to deal with. But I think as you find as you go through the different publications, what you'll find is a lot of these implications are already occurring. These are happening in the amplification, not in MinElute post-PCR cleanup. So we really have to separate out where these different issues come up. And then we talked about implications too before. So let's say we learn about MinElute today. We want to apply MinElute in our laboratories, and then how do we take that back to the laboratory? How do we get this? We need to validate it, certainly. There's competencies. We have to ask, is this going to be a routine task, or is this going to be a specialized? So we have someone who specializes in it. Do we make it the entire laboratory is responsible for understanding this? So again, just different things to think about, and also setting up the laboratory to be able to handle it. You know, you may want to set off a small closet to work on these type of samples. Again, it's in dealing with these low template, especially low template amounts. I know you all have various laboratory setups, and so we have to think about that setup as we implement it. So again, now my main objective really is to get into what is the post-PCR cleanup, give you a little bit of the idea of the technology, why it works, how it works, and so we'll do this over the next couple slides. So again, in terms of silica membrane type technology and silica gel technology, it's been used and documented for purification right from an actual sample type. So certainly it is an alternative with different types of technologies outside of the post-PCR cleanup for extraction from primary samples, hair, cigarette butts, chewing gum, and various other types of samples. But then also the type of technology can be used for extraction cleanup as well. So we've seen application of our Kaijin, for instance, technology used for cleanup, as many people use the microcon columns for for PCI cleanup. Same way, Silica Membrane and Silica Gel Membranes can be used. And then again, post-PCR cleanup, and that's really what we're going to highlight today. So again, the goals of, from really, I would say, our perspective of how we can support the forensic community here is to and is to really increase the sensitivity of the alleles detected. And so again, and I think Rob will probably get more into this certainly for you guys, is now that we have this new technology, how do we implement it? How do we create these new STR interpretations? And there's some things we need to think about downstream. And also, what are the benefits for, say, mixture analysis? And we'll talk about Foster et al. And how some of their work really showed the difference between doing, say, 34 cycle PCR and using MinElute, and its ability in their mind to be able to do mixture analysis in a much better case than by doing 34 cycle. So we'll talk about that as well. So again, preserving the sample. So again, in some of these different types of workflows and the different types of technologies, and one of the things we want to try to do is to eliminate, or we want to try to maintain the type of workflow that we have currently. So if we have standard volume, standard cycle, PCR, let's try to find little tools to tweak the system and to make it work a whole lot better than it has in the past. And so that's really what we've been able to achieve with MinElute. If you want to look at, there's a lot of expensive techniques out there, like say laser capture microdissection. If you look at it as an alternative for enrichment, because that's really what we're doing here, is we're enriching the target with MinElute. And if you look at other techniques that can be used across the workflow. So again, this is more in your extraction laboratory where this type of technology is used, but this is inexpensive. I think microscopes run from anywhere from $100,000 to $200,000. There's very intensive training, and it's quite a long process where we could look at implementing a 15-minute protocol with minimal validation time with technologies like MinElute. They complement each other. Certainly they have a place to be used in the same laboratory, but again, sometimes we can keep things simple, certainly with using technologies like MinElute. And then the last goal certainly is to make things just automation friendly. And tomorrow we'll really get into the application of automating MinElute on our QIAcube system. So Silica Gel Membrane technology, what is it? What does it look like? So you'll see this in the laboratory later on today when you start doing some work. And again, what this is, it's a smaller membrane that we offer. It's the pore size is a little bit different than some of our standard membranes, but really what the MinElute really has to talk about is the size of the membrane. And what I always tend to talk to people about or relate it to is the whole needle in a haystack. So if we had say a binding capacity of our of a general, let's say our QIAmp mini column. QIAmp mini column I think has probably a bit over 10 micrograms of total binding capacity. Well, if we have let's say 50 picograms hidden in that big membrane, it's harder to get that to come off of that big membrane. So really the nice case of why MinElute works, and then that's why we also recommend our DNA Investigator QIAmp product, because it again utilizes that MinElute membrane. We don't have, I think more likely forensic scientists is scared of having 10 micrograms of DNA on an extraction. But in the case of where, again, that whole needle in a haystack, we want to pull that little bit of DNA out of a much smaller haystack if we can, and it makes sense to use a MinElute membrane. So again, how and why does Silica membrane? Now, some of you, I know from looking around the room, have our EZ1 system, and so this would be the same thing whether you have a Silica membrane, Silica on paper membrane, or if you have silica wrapped around our magnetic bead-based system. Basically silica is silica, and what we do at Kaijin is we utilize the same type of membrane, same type of silica, and it's just on a different coating, and then we also use the same type of buffers. And you'll often find, for instance, AW1, AW2 that's used in both our QIAmp product as well as our EZ1 product. So as you see it here on the screen, the same mechanism works across. And again, I pictured it this way because again, this is I kind of tilt it sideways into a chemical equilibrium experiment, really what's going on here. And what we're doing is we're absorbing under a high chaotropic salt, lower pH, and we're adding a alcohol. And so this creates the binding capacity. to get the nucleic acid to jump onto the silica. Now what we will have, and what's going on, and we'll talk about here in a second, nucleic acid isn't the only thing that wants to jump onto the silica based on the structure of it. We have proteins, we have a lot of different things going on. We also, the way we deliver these products, you have protein that can attach to the column and other types of things. We see a lot of carbohydrate proteins attached to columns, for instance. So as we go through the wash process, that's how we get these different would end up being PCR inhibitors out of the prep, and we get to a very, very concentrated and very pure sample where we elute with no chaetrope. We get rid of that chaetrope. We're under a higher pH and a low salt condition, and we'll see that here in a second here. So again, what you can see here, we have water molecules that are wrapped around our DNA sample, and so silica, as you see there, is SiO2 is pictured there. And really what we're doing throughout this process is, within the lysis and binding process, is to introduce these chaetropes. And these chaetropic salts, what they do is they really create chaos, is the best way to say it. When I first heard it from our training group, it really started to make a lot of sense to me. And that's really what we're doing, is we're stripping, what we're going to end up doing is stripping these water molecules off of the DNA, and DNA just wants to be protected. So it's going to go where it can get protected, and that's onto the silica membrane or silica magnetic bead. Again, this is occurring in both cases. In the case I just want to mention here, the carrier RNA, this is a general, what I wanted to do is to show a general extraction process. Now, a lot of our protocols on the DNA extraction side, not necessarily cleanup, we suggest using the carrier RNA. And the reason that we do that is, again, as I showed you in that last slide, we're talking about an equilibrium. And it doesn't take a lot to start and to initiate that equilibrium such that that DNA is jumping onto the silica and attaching and binding to the silica. But sometimes we need to give that equilibrium a little bit of a push. And so we add the carrier RNA. It's a synthetic oligo. It's a sugar backbone based. And it's just a random repeat of oligo DTs type random primers. And so And as you'll see throughout this process, they are cleaned away typically within the process. When a lot of these hit the chaetrope, they actually break up and decompose. But I wanted to show the carrier in this process as well. So now that we have our DNA binded to our silica, now we want to go through the process. So we talked about before is now we want to wash away with different amounts of chaetropic salt. So these chaetropes help break up. So if you want to figure out protein, so let's relate this to post-PCR cleanup. TAC is a protein, or equivalent of basically a protein in terms of this process here. So what we really want to do is to decompose really the protein, break it down so that it's not binding to that silica, because again, they have different properties that are going to want them to grab onto silica as well. I mean, for instance, we have protein extraction columns as well, and we use silica. So obviously they want to bind. And then we also have different types of inhibitory contaminants there as well. So you'll often see that we want to get rid of those, and then the carrier RNA. So again, as we go through this process, we want to wash away with those chaetropic salts, get rid of those proteins, the inhibition contaminants, and then the carrier. And next thing that we want to do is we have now this water, this chaetropic salt that's still there, and obviously now the funny thing about this entire process is every single step that we do is really goes against what you all would like to see in terms of getting an eluted nice DNA sample, because these same salts that helped us get rid of all these contaminants would now inhibit our PCR. So now we need to take care of those. And so we do that by washing those with ethanol. That's going to help us get rid of those chaetropes. And so that leaves us with, as you can see here, just our DNA sample. But then again, what's not good for PCR? Ethanol. So what we need to do is to dry that ethanol off of the sample. This was the best way I could show without, there's no videotape of a centrifuge, so that was the best I could do really for you. But really what we need to do is we need to dry off that ethanol so that, again, it's not going to contaminate or inhibit, I should say, really our PCR downstream. So now we have our silica, we have DNA binded to it, highly purified DNA on it, and now we need to get that DNA off, which is obviously just as important as getting the DNA to bind. It's just as important to get it off. So we do that again with a low salt buffer and a pH between 7 to 9. So the various technologies, and we'll talk about MinElute here in a second, the specifics, but again, for the most part, we're in a pH range of 7 to 9. And in terms of our elution buffers that we utilize for the forensic community, it's certainly in line with and has compatibility with all the STR kits, all the quantifiler, plexer chemistry, all of the different quant technologies. So it's a low TE based buffer basically that's used for the most part. So what we're doing is we're basically reformulating this water. We're creating conditions so that that DNA can pop off of the silica, and so that's what the low salt buffer and the pH is described. It helps that DNA pop off, and that's very important. Now, within some silica membrane type technologies, we'll see some people, especially on the extraction side, like our Kaijin customers, they'll do a double elution process, and again, that allows them to often to also add really some agitation to the process to try to get that DNA off of the silica by running 2 elutions. Some people reload the elution, some people split it in half. So if they're eluting in 20 microliters, they'll run 10 elute, throw another 10 microliters of buffer and elute, and capture it all in the same tube. So we'd really have to do probably a longer term study to figure out how well that works and what the cost and benefit of it is. If some people want to go back and take a look at this process in terms of an academic paper, this is really just a listing here of one of the researcher up in Canada who had done some work to understand what's going on in a column. So this was after, and why does this technology work the way it does? And so again, I suggest that you give it a read. It sets, or at least put in your binders for people that do incorporate either this or our other technologies. It's a good read to keep in your developmental validation and validation processes. So let's talk about MinElute in particular here. So these are the kit contents. So there is 2 different formats. There's a 50 quantity kit and there's a 250 quantity kit. And Kaijin, we do offer, there's a little bit of buffer extra. There's usually about 10% extra buffer that is required. So you're looking at these buffer PB. That really means nothing to you at this point. But what that is that is our high salt binding buffer. Buffer PE is the wash buffer that you can see there. It says concentrate because within the protocol, as you guys will see, is we add ethanol to that, and alcohol to that. And then buffer EB is the elution buffer, and that's it's a 10 millimolar tris CL, and it is at a pH of 8.5. And so that is the elution buffer. Again, this helps with this, or it works well with the downstream application of the STR. And at this point, again, we've already done our amp, so it's really compatible with, in this particular case of MinElute, it allows for compatibility with all of your reagents that are going to go into your 310s, 3130, 3130 XLs. So the procedure. So there are various ways to do the procedure of MinElute. We have and I'll kind of move from right to left here. We have a vacuum-based protocol, so you can start by doing the first couple steps on a vacuum. It's an easy way to do it. Again, I would say that this would probably be more for research applications, why this was developed. Again, people doing sequencing and resequencing work. In a forensic laboratory, the one problem that you run into in the case of trying to do the vacuum is vacuums are, I would make sure if you were going to do this process that you use a microprocessor controlled little pig vacuum, as they're often called, the pig vacuums. Just make sure that it's not like, say, a house pressure, because that can vary quite a bit. And so if you are going to use one, make sure that it's one supplied by the company, because you will get better performance. It'll make sure that that pressure stays maintained while you're doing that and pulling that vacuum. The other way is, which especially probably go through today, is the centrifugation process. It's probably recommended. It's easy, as you guys will probably see today, and with very little manipulations, again, within the centrifuge, it's probably recommended. And again, the final one, and we'll get in a little bit more detail with that tomorrow, is the use of the Kaijin. So the Kaijin is just quickly, is a automated centrifuge that we launched a couple years ago. It has a robotic arm, and it has slots for 1 to 12 samples, and that automates about 100 different of our protocols that we offer at Kaijin. So our Chi-Amp's offered in there. Our protein, our RNA work is all covered within the applications on the system. And obviously our MinElute cleanup is as well. So getting into a little bit of the technologies, we've also had some inquiries from customers. Which kit do I use? So a lot of people have heard about the kit or have read some research, and so they say, okay, well, I went on your website and there's a couple different things with MinElute on it. So again, remember that MinElute refers to just simply the type of membrane. And then you can look on the back there, you have PCR purification kit, gel extraction, and then we have reaction cleanup. And so they're all a little bit similar, but they all have different purposes. And so in the case of the, say, the reaction cleanup, that would be for different types of enzymatic, for people looking to make certain cleavages, and you want to get rid of that enzyme or something. That's what that reaction cleanup is really used for. Gel extraction is just what it sounds like if you were doing a gel to do some separation. But then the one that we really want to concentrate here on is the MinElute PCR purification kit. And you can see here the different area of the fragments removed and then also the fragments recovered. So why this kit has been beneficial really is, as you can see, it basically does not purify anything below. It's hard to really measure because these are so small fragments of DNA sometimes. It is hard to give you a solid number in terms of how much is or isn't. But what we do know is that the way that the technology is set up is that it really allows for purification of 70 base pairs or greater of DNA. And so that what you're going to see is that, and we talk about the different things here, is that all these primers and all these different DNTPs and things that are left over, that's what's really going to get cleaned up here, and that's what allows for it. So if you look at the MinElute kit here, the specifications really, we see that we have, again, a 5 microgram total binding capacity. Now, again, the reason we use the MinElute is because if this was the regular membrane, you'd see anywhere from 10, and some of our kits go up to about 20 micrograms. But again, that's what we were talking about before. You can see that elution volume can go down to 10 microliters. Some people choose to do a little bit higher. We wouldn't suggest, certainly, to make sure that we can fully wet the membrane, you don't want to do anything less. And then you'll see there, based on the typical centrifuge recommendations that we make, you'll get 9 microliters out of it because the membrane will retain a little bit of that liquid. So again, and that's why you see the recovery of the DNA around 80%. Because a little bit will eventually get trapped. But again, if we're talking a PCR product cleanup, we typically have enough components left over. Again, you can see the recovered amounts, and then also it removes everything less than 40 mers. So getting into why do we see the results that we've seen from this technology really is what this slide shows here. So in our typical capillary system, we've done our amplification, and what is left in that amplification is seen here. We have primers. We obviously have our DNA there, DNTPs, salts, and if our amplification didn't go well, we'll also see that's where we're seeing dye blobs, and we're seeing some other things left over from that amplification. And so when that then goes up into your capillary, what's going on then is really that as we all know, and focus is really the word that comes to mind here, is that the electrode is focusing power on things other than what we want to look at. So that's really the concept here, and that's really where we go. And you can see here the DNA gets cleaned up throughout the PCR purification process. It only leaves us with what we want to look at. So that electrode and really creates a optimal situation really for the capillary to look at what we just want to look at, which is the DNA. So now what I'd like to get into, and again, I kind of talked about and alluded to before, Kaijin is a purification and assay company. We've been providing MinElute for quite a while and its use for different types of applications. But then this is really where we either collaborate with our customers, or our customers certainly take things in their own hands and do some studies with the kits. And so this was really one of the first publications where people have shown, and I can tell you it was used before, in New York City. They had used a different type of kit, one of our DIEX products. Again, this was more from sequencing, but they used it for a lot of their World Trade Center type sample cleanups. So it's been done before, but really to go into a research environment and studied, this was the first paper. It was Smith and Valentine, and this was simplified low copy DNA analysis by post-PCR purification. And so within the process, and I think all the papers are either available to you, or you can always order them off our website, but I believe all the materials are available to you here. But again, this was JFS, and so I know a lot of you have libraries too, so you can get back to your laboratory and take a look at it. Again, here, Smith and Valentine demonstrated really for the first time its use in application. They looked at it for, again, their target was that low copy number type template of less than 100 picograms, and they compared it to the alternatives, which again, as we talked about before, is the nested PCR, low volume PCR, and increased cycle number. And so that's really where they looked at. They did use phenylchloroform as an extraction process. They also, within it, they did just some general research studies of it, and then they did some mock samples. They did a hair shaft, they did a dermal ridge fingerprint on some glass, and then also on some paper. Excuse me. So then they really looked at the application of it as well. So they looked at it from a developmental process, and then also trying to do some mock casework with it. I do want to point out that as we go through, some of the data that you will see in this process was done by QuantiBlot. So at the time of the research. So we all probably know that the values are going to run, the quant values that you expect to see are going to be really, we want to times those by 2, 3, right around there is typically what the conversion is. So again, look at this from a relative standpoint, I would say, but it'll start making sense to you as you also see the numbers. You know, we'll see detection down to, say, 5 picograms. That I think more in line with, you know, around 15 to 30 picograms, things like of that sort. And you'll see that data. One of the things that they investigated is allele drop-in, drop-out. Look at the effects of the process versus using just standard processes, and also the 34 cycle, looking at the stutter, looking at possibilities for contamination. They did see in their particular case one case of contamination that they were trying to figure out, and it's noted in the paper. But again, this is always something, whether you're using MinElute or not, if you're doing these type of low copy, low template amounts, obviously we need to take proper precautions. The Foster et al. Paper and study that was done, and we'll talk about here too, again, they saw a little bit, and also, you know, they did double gloving, they did cleaning of gloves, they top to bottom. So I think as forensic scientists, you all know that the use of the application of this type of technology really pushes the limits, and we have to be very careful at the same time. So within this Smith and Valentine study, the one thing that they did do was to they did do some comparisons of other technologies. So they looked at Silica Membrane with the microcon, and the montage chemistries, I think by Millipore. And then they looked at an enzymatic process, the exosapit product, and then they looked at the MinElute. And as you'll see in their conclusions, really, that one of the things that they saw for the time, the amount of time or the small amount of time that it takes to do MinElute versus the results that they did see, MinElute was their suggested tool to move forward. The exosapit really just had created actually some greater problems, which they ended up actually cleaning up with MinElute, and then it worked well. So it really shows you the application of the technology of MinElute. So again, let's talk a little bit about what it does. And so this was a analysis of this was the hair sample, I believe, where you can see here the noise that we see in the various levels here, the various channels. And this is a purification without using MinElute. And then really what we can see here at 20 picograms. And then once we do clean it up, you can see the application of it. We're reducing that noise. We're getting a lot of that noise is created from those DNTPs and those salts. That's really what that noise is seeing. You'll see other applications in the use where we get rid of, you can really notice those dye blobs go away. I believe a lot of people see that mostly in the VIC channel, but I've seen a couple different things. But we'll see different. You can see different applications of where those dye blobs really went away as well. So again, what we're doing is you can expect to see, and one of the things that also Smith and Valentine talked about was they also talked about concentrating the sample as well. So again, with the use of this type of technology and its recovery rate at the different volumes that it allows to elute, they were also able to, and they ran an experiment to inject the entire 10 microliters. So with the various samples, they looked at the effects of also injecting all the amounts of the full amount of DNA. So again, they did show that they obtained data down to 5 picograms, again, quantiblot, so just take that into small asterisks, I guess. And, but then I know that Pam, in particularly, when she got to her new position in Texas, she basically revalidated and reran this experiment as well with QuantiFiler and some of the technologies that they had within her new laboratory. So, so again, she found basically the same findings, just with the different scales of using QuantiFiler. Is basically. Again, when injecting the total amount, again, you can see they saw it to a 19-fold signal increase. Again, you'll also go through as you read this too that you will find that there was, when they did inject the entire amount of PCR purified sample, that there was some increased stutter, I believe at D5, that they did see. And so there is a study that there's probably just not enough time to go through today, but I highly recommend you go through the paper, is that you will see some increase in stutter as you go through and inject that entire sample. But if you follow a typical protocol, follow your template as again, with the idea of utilizing this as a tool and then just using your identifier or your PowerPlex 16 as is, then again, we can see some of the gains within this technology. Supporting journal article, what I wanted to just put this in, certainly a lot of the interpretations and need for interpretations and the whole concept of now low copy or low template comes out of this gentleman, Peter Gill. So obviously a good read. I'm sure a lot of you have probably already gone through this, but send all these emails to Peter if you want any ideas. He likes exchanges of comments. So he's obviously a thought leader, I would say, but obviously those come with a lot of different perspectives from various people. So now what we want to do is to get into a couple more papers here and a couple more applications. So again, Smith and Valentine really kicked things off, and really what we did here was to take that application and try to get in more mock casework samples and so on. I'm going to advance one here. Basically what we're doing is we're going into the Foster et al. Here. But what he wanted to do was Foster et al., what we saw was Post-PCR Cleanup. They saw, based on some of the initial publications, that there was some definite purpose for using the Post-PCR Cleanup. They also, in looking at some of the work by, and some of the publications, some recommendations by, I think it was Bedoli, and then also just taking into, I think it was Wickenheiser, had done, in some of their work, had really shown that the success rate on typical routine forensic processes will give you a total success rate of 30 to 50%. And they really saw a need to really increase that, and that's the application here. And so they saw a use for the post-PCR cleanup. And then also, they also would really try to utilize different types of technologies, or I shouldn't say technologies, but derivatives, So changing injection volumes and changing the kilovolt on the electrode and such. So again, what they really wanted to look at was a comparison of making these slight modifications, what to expect when doing 28 cycle, what to expect when doing 28 plus MinElute, and then also 28 plus MinElute when we change some variables like injection volumes, and then also rating it against 34 cycle, what most people put into that realm of LCN type applications. So you'll see in the results that again, what they found ultimately was a combination of the 28 cycle PCR combined with some variables. And what if you go through the publication in detail and some of the recommendations, there's what we're trying to do is to run this, it's a cost benefit of using too much sample and not consuming too much sample. But the same wise, you can see that they suggest too is this can be a stepwise approach. So we can try our standard 28, we can then add MinElute, we can then also change injection time. So I think that's one of the things that the paper best shows, I really feel, is that this can be a stepwise if we had enough sample. Now again, if we look at why are we using MinElute, it's typically with LCN or just low template, low evidence amounts of sample. So there's that cost benefit we really need to think about. And again, that's when you go home to your laboratories, that's one thing that we need to really think about is that process there. Again, one of the things too that they did was they did do some consensus amplifications too. Now they did this for the purpose not only of just some of the work I think that's been done by New York, but also to really rule out any and to really measure some of the drop-in dropouts that they saw. So by creating these consensus protocols, or profiles, they also were able to better measure the drop-in dropout of the profiles. And again, what they did see was that, and really one of their main recommendations is by utilizing MinElute and with some of these tweaks in the injection volumes and the electrode, what they did see is that certainly it'll outperform the 34 cycle and create less issues. But then also for mixture analysis, they felt that MinElute plus some of these other variables will allow for better mixture analysis and won't inhibit some of your mixture analysis. And so again, as we go through, I highly suggest that you go through the entire paper in its totality. Again, here in A, what we can see here is, so the MinElute type, the processes that have MinElute are in B and C there. And then you can see that some of the variables, we have 30-second injection time as a variable here. You'll notice that one thing to just look at what's being done, we have 2 and 2 here for injection volume. So that part, when MinElute was utilized, they were using different volumes that were used in both of the processes. And then the other variable here was the injection volume, 4 kilovolts here when they use C. So again, one of the things that you'll notice is that when we go from A to B, we're looking at obviously you can see just that is the main process that we want to see of just cleaning up the sample. So again, we're cleaning up the salts, cleaning up the DNTPs, and it's giving us better profiles, better baseline, better peak resolution. The longest peak, if you look at it, really goes from about 60 RFU up to approximately 500 RFU. So again, that's the type of things, and you'll see that, I think, when we go through the different lab practice, that's what you'll see, and that's, again, another thing for interpretations that we need to think about moving forward. And then when we go from B and C, again, when we change the injection time from 10 to 30 seconds, we change the injection volume from 3 to 4 kilovolt, again, you saw the largest peak go from about, again, that 500 RFU up to about 3250. So in this particular case, and they note this in their paper, is that it really maxed out their system from where they were analyzing. And so, so again, it's just things that we need to think about as we go through this process, and also that shows sometimes the need to either concentrate or not concentrate. So those are the type of things that you have to get used to as you utilize the kit. And again, in their assumptions, both B and C would be good in their mind in terms of mixture analysis as it relates to LCN type applications, or I should say 34 cycle applications. And again, this really highlights it here. So Kathy Mann's press came out of Jack Valentine's laboratory and continued a lot of the work, and this was some of her research that she was doing with Jack, I believe, at the time. And what they looked at was, again, and we talked earlier before about the use of MinElute can be a complement to other types of technologies, and this is a great case of it, where they were using YSTR from a couple different companies. They did the Y-Filer, PowerPlex Y, and then a kit from Reliogene. And what they really were looking at measuring was postcoital samples that were taken, and it was a small study. Certainly, when this was presented, they said they could certainly benefit from a larger sample pool than originally was there. But again, collected after intercourse, and the time points were, let's see, time points were 72, 96, 120, 144, 168 hours after intercourse. They did use phenylchloroform extraction. The other thing that they looked at too was the use of either differential or not doing non-differential. I know that there's some laboratories out there who are just taking those swabs, doing a full extraction, doing a Y quant directly on their first swab to triage their sample. Is there presence, not presence of DNA? So this also shows the application of that example. It's more in the Y quant where some people are doing that, but they showed the difference of doing the differential or not doing the differential. When differential was used, they saw partial profiles, and then when the MinElute was used, they saw better performance overall and a total increase in the loci that were identified. So again, what they saw in the second point here, in most cases, they saw an increase in total loci that were analyzed or able to be analyzed, and so that was a definite benefit. As you can see here, with the increased peak heights, they were able to get partial profiles where they weren't before. And in greater detail, they were able to, by using MinElute and some of the commercial kits that are out there, they were able to go out to 6 days and obtain a partial profile 6 days after intercourse from these cervico-vaginal swabs that were taken. Again, just really show the application too of just using the YSTRs with MinElute and increase the sensitivity of these sexual assault samples. Now, when Kathy left there, she took this application over to Arizona, where she's I believe a CRIM over there, CRIM 3 maybe. And so she took this and said, okay, let's take this to the next level and try to get some application, some real application data out of it. And one of the things that we do see is that again, she saw, what the nice thing was, she saw the same thing that she saw in her research and started putting this into its application. They showed it and they compared it to other methods, and then also what they wanted to really look at was some very difficult samples. I can say that they've put this into routine application, and actually they had a serial rapist who was recently admitted to crimes because of this technology. They one particular sample they used MinElute on, gave a profile, they came to the guy with the evidence, and he admitted to it, and I think it was several rapes that he was convicted of. So it's certainly getting put into application as well. Going into another study here, we talked earlier about how and why, when to use these type of technologies, and again, we're getting into very tough sample types. I know that a lot of you probably know Pat's has certainly has been known for his work with bone samples, and I think that was widely utilized. With Katrina and some of the things, certainly a lot of those bone samples were coming out of the ground, and so there was a great importance there, and they do a lot of work at North Louisiana on that. So one of the things as they were doing their developmental validation of the technology, you'll see some slides here as we go through. They just want to say, okay, how does it look with a standard, and then how does it look with our sample? So these are the conditions that they used. So they had 3130. They were analyzing at 150 RFU values on their GMapper, and then again they used just our standard out of the box, out of the handbook, I should say, protocol for the MinElute cleanup, and they used the 10 microliter elution volume. So as you can see here at the bottom, you have you have here, this blue line here is the, you called it unclean, but that's the unpurified, so just a standard amp. This was the performance of the amp here. And again, what you have on the, going up the side here is the average total RFUs. So you can see a nice linear value here when we used a 1-to-1. So that 1-to-1 is basically where you have a 25 microliter reaction and then you elute it in 25 microliters. So we talked a little bit before about the cost benefit of concentrating down to 10 microliters. And so you can see a little bit of that here when you look at this, what they call the 2.5 to 1, and that's where she went from the 25 microliter extraction process and then purified. So you can see that really there's a cost in terms of total fold. You kind of see this plateau, and that's what you really could expect. I mean, the system's going to max out on a lot of the capillary systems and the way that, excuse me, that they're set up. You can see here that she used the 9947A standard, and we'll see a little bit of that here in a sec. And then she used 1 microliter of product when she went into her amplification. So when they were doing their developmental validation, they again just looked at the standard, and this is 7.8 picograms of that template going into a Powerplex 16. You can see here the with and without that's seen within the MinElute chemistry here, with and without. So in some of these cases in particular, I mean, she already highlights them here in red. You can see that, again, there was just noise. We weren't getting good. This is a perfect example of where that electrode and that focusing power of that electrode in the capillary weren't able to detect that DNA that was going through, going up the capillary. And so it's a perfect example. Clean up that sample, allow that, the power of those instruments to focus on the DNA, and then you were able to capture that nucleic acid. Now, looking at the bone in particular, you can see here that we went from, you can see that the scales definitely increase. Her findings in general were that without MinElute, she had about a 26% call ratio of her allele peaks. And then when she added MinElute, in all cases, from the bone sample that they expected, they got a 100% call rate when they used MinElute. So again, looking at some of these difficult, this was the pulverized bone sample, definitely can help out. And in most cases, they saw about a 7-fold increase in the performance of the total RFUs over the others. And again, in some of these cases, we just had no calls. I mean, if you look at, again, in this area here, we see the calls versus none. And again, the data, it's a little bit fuzzy on here, but again, you can see there's about anywhere from a 4 to 7-fold, but mostly they saw a 7-fold increase across. And again, their results that they saw here is that you can see the typical increase. Average RFU is 241 to 16, just over 1,600 RFU. And again here, whenever MinElute was used, they saw 100% alleles were detected as were expected. And obviously, you know, I think a lot of people are running into these type of difficult samples, bone samples, hair shafts. And I know some people I have worked with actually in Tijuana have a lot of bones that they're dealing with, and they're going to be looking at utilizing this technology. The scientist down there has something like 3,500 bones that need to be processed, so from the drug wars. So certainly major implications there. Give a lot of people some closure, hopefully. I won't go into too much of this detail because Robert's going to go into this, but we can see here just, again, average signal increase. And these are all things, it's not that it's great that it happens. We need to think about what the implications of all these things are when we put this into the laboratory use. But Robert will go into more of this detail in his study, looking at multiple downstream STR kits and its application there. Again, really what we want to do here from our perspective at Kaijin and trying to help out the forensic scientists here, we really feel that this technology can be used to enhance your applications and with minimal amount of major impact on the laboratory. So again, we talked about, for instance, other alternatives like laser capture microdissection. It could probably take a full year, if not more, the way I've seen some of these labs take on new technologies and validate them and create competencies. Versus MinElute, we've seen customers get online in anywhere from 3 to 6 months. Again, we look at the cost benefit again of doing these type of technologies for 75 cents or whatever it is versus what's involved with the laser capture system. Again, they can complement each other, but it is a way to look at utilizing and do little things to your workflow. Obviously has implications in low template DNA samples. We're seeing a lot more touch. There's a lot of breaking and entering type samples coming in. Some of those are fingerprints. Some of those are blood on a piece of glass. So obviously they have, and we need to think about how to triage those different samples because they're going to have different ones. You know, the blood sample is going to give you a lot of DNA, the other's not. And again, one of the things we want to do is look at this use versus other types of technologies. Your nested low volume PCR type technologies, 34 cycle and such. And also we want to make sure that it's automatable as well. And again, we'll talk about that in a bit. And finishing up here, the last piece here, we did do a webinar. I'm looking around the room at some of the faces, and I think some of you were on this, some of you weren't. We did do a webinar. We had New York State Police who have validated the kit, and actually Jay Caponera has, he just gave a presentation up at the Green Mountain DNA conference up in Vermont, and he showed a couple cases where they've actually put that into application. Right now the big thing that they're working on is their interpretation rules. But in terms of, again, him and another colleague being specialized in it, they've put it into application in a couple cases. Jay will talk to anybody about it, so please give him a call. And I think I've also sent the validation paper to Robert if anybody would like to see what they did to validate the technology. And then Bruce Heidelbrecht from Maryland State Police, they've looked at implementing the technology. They have a Kaijin. Jay is doing it currently manually, and Bruce is doing this with the use of the Kaijin instrument. So again, I think you see, the nice thing is you see the same findings and the same concerns, drop in, drop out, stutter, and so on. Again, things that already are happening within low template DNA applications, but then again, you can see the difference between automating and not. And then there are some YouTube videos that I think our marketing department put together. Actually it was just one of our application scientists that was doing, just showing how easy it is to use MinElute in the laboratory. Thank you very much.